everyone, this is Suman, and today we are going to learn about a topic that is called counter current immunoelectrophoresis. It is a type of immunoelectrophoresis, but there is a modification. Generally, in the electrophoresis, the movement is from one electrode to another electrode. Suppose from negative electrode to positive electrode, or from positive electrode to negative electrode. But in case of counter current immunoelectrophoresis, the antibody and antigen they move towards each other. That's why the name is counter current. So you can say it is a rapid portion of double immunodiffusion. We have already discussed about the double immunodiffusion where the antigens and antibody they diffuse towards each other. That's why the name is double diffusion, as the antigen and antibody both have the ability to diffuse towards each other and form a precipital line. So let's discuss the counter current immunoelectrophoresis. As I have said, the name is counter current because one charge is prevailing the other. That means the negative electrode moves towards the positive electrode and the positive electrode moves towards the negative electrode. So as we have discussed in all the immunoelectrophoresis process, first we need to make the agarose solidify it. After the agarose gets solidified, then you need to make two wells, one for the antigen and one for the antibody, by the gel puncture without breaking the total agarose. So after that you need to pour the antigen solution in one well and antibody solution in other well. Generally, the antigens, the most bacterial antigens, they have negatively charged. We all know that. The capsular polysaccharide, as the capsular polysaccharide is slightly alkaline in environment, so the antigen carries the negative charge, and the antibody it is less negative. That's why it is positive charge. So this is the electrophoresis tank. So you can see a precipitant line is formed. So the antigens and antibody they move towards each other and form a precipital line. So the antigen is here unknown, but the antibody is from a known specific sample. Okay, and they both are applied in the electric field so that they can move towards each other. Generally, it is a qualitative process, as we are not discussing about the concentration of antigen or antibody. But if you see the thickness in this picture, the thickness is slightly more. So you can say that the antigen concentration is more in this experiment. If the line is very thin, then you can say that the antigen concentration is less. And if the line is not at all visible, then you can say the antibody is not present for that particular antigen. So let's move to the functions. The functions are it generally helps in the hepatitis B surface antigen detection, and again in the detection of the fetoprotein, and it was used for the rapid diagnosis of some bacterial, mycoplasma and viral infection. So this is all about the counter current immunoelectrophoresis. Let me summarize it once again. In counter current electrophoresis, it is same as the double immunodiffusion, but it is a modification of that, as it is related to immunoelectrophoresis. So if the thickness of the precipitant line is more, you can get some measure of quantity. So then you can say that it is a quantitative method, as the thickness is directly proportional to the antigen concentration. So the moment is the antigen is, as it is a negative charge, it moves towards the cathode, and the antibody which carries the positive charge, it moves towards the anode. So the flows of the charge revealing one another. So that's all about the counter current immunoelectrophoresis. If you like this video, please hit the like button. Do subscribe to my channel to get more such videos. Thank you.